Okay, welcome everyone to this spring SWOG meeting in beautiful San Francisco. Well, just kidding. I guess I'm one of the few here, but maybe in the fall in Chicago, we'll see. But thanks for joining us. We have a very full agenda, really an exciting time, I think, for the Leukemia Committee with current and especially upcoming studies. So just briefly, we'll show you the agenda. Next slide. We'll start, Dr. Erba will give a brief introduction, and then hopefully if Dr. Stevens has been able to join, she'll start with CLL. Otherwise, we'll launch straight into AML. Next slide. We also have a big portfolio coming on board for ALL. Really, I think a renaissance for this oft ignored disease. Next slide. And then Jerry will give us a presentation. Always looking forward to Jerry's talks on translational medicine, what's happening. Gail and Bruce Wright will give us a brief update on patient advocacy, a increasingly important part of each and every one of our studies. And then Dr. Sweet will give a brief update about her ongoing study with CML, and then we'll wrap things up. So I see Dr. Stevens has joined. So let's turn the agenda over to Dr. Erba for a few words, and then we'll hear about CLL.
Thanks, Steve, and thanks all for joining. First, I want to thank Steve. Not only is he the vice chair of the Leukemia Committee and helps me get through everything that we do, but he's been instrumental and has taken the lead on organizing our spring Leukemia Committee. So I really thank him for doing that, along with Courtney and our operations office. I'd like to introduce quickly some of the leaders of our committee. As you know, I'm Harry Erba. I'm the chair of our committee. I'm at Duke University. And Steve Coutre at Stanford is our vice chair. Jerry Radich is the chair of our Translational Medicine Committee. We have our executive officer, Susan O'Brien. We also have Gail Sperling as our patient advocate. In our operations office, we have Melissa Biddle, and Sandy Hida continues to help out quite a bit. And on our in our biostats, the head of that group is Megan, working closely with Anna Mosley. And then because I've been really focusing a lot of my attention, not just on the entire committee, but really focusing a lot of my attention on the initiative that you're going to be hearing about, MyeloMatch. I felt it was important to have leaders in our committee that took leadership and direction for subsets, and currently that is Steve Coutre for chronic lymphocytic leukemia, and it's Anjali Advani for acute lymphoblastic leukemia. So appreciate, I think I'm doing all this from memory, so I hope I didn't miss anybody. If I did, my sincere apologies. You know, I'm not really good at scripting things. It has been a really productive 6 months. I know our portfolio has been sparse, but there's a reason for that. And many of you know that we have been collaborating very closely with our colleagues in the other North American cooperative groups that have leukemia committees. So the Canadian Clinical Trials Group, the Alliance, ECOG Akron, to develop clinical trials in chronic lymphocytic leukemia, clinical trials in acute lymphoblastic leukemia, and in acute myeloid leukemia and myelodysplastic syndromes. And so we're kind of redoing how we have developed protocols everywhere. It takes time to get that new portfolio up and going. But I think you're going to be impressed by what this group has done in the last 6 months. So want to just start by thanking everyone for their hard work in doing this and helping people with leukemias and try to achieve a better quality of life and better outcomes, which is what we do every day at our own institution. So with no further ado, let's turn it back to Steve and he'll introduce our first speaker.
Great, thank you, Harry. And I want to second your comments about the support staff for the Leukemia Committee. Absolutely amazing individuals who make our jobs so much easier every day. I also want to emphasize that we are fully committed to involving young investigators in particular. We want you to take leadership roles in the areas that you have interest, and we're committed to advocating for SWOG leadership where appropriate in the intergroup studies. So for those of you on the call who haven't had the opportunity to lead a study or design a study, if you have interest, please reach out to those of us within the group who have led some of these areas because that's really our goal. I certainly don't anticipate being the PI of any study in the future within SWOG. I think it's more appropriate that we try to help develop everyone else's careers. So let's turn to our first topic. That'll be chronic lymphocytic leukemia, and I'm happy to introduce Dr. Deborah Stevens at the University of Utah, who's been leading our efforts here, along with several other colleagues within the intergroup, and in particular, a newly open study led by Dr. Stevens. So Deb, take it away.
Thanks, Steve, very much for giving me the opportunity to talk about the CLL studies today. I really want to focus on our own study, the SWOG S1925 that just opened up, but I am going to give you guys some updates on the ECOG and Alliance studies that are currently ongoing as well. You can go to the next slide. You can go ahead. So in general, the overview of these studies, and I'll get into more details of these as we go through the presentation. So our study for SWOG, the S1925, is really for frontline asymptomatic patients. If you want to go to the next slide. The ECOG acron is frontline for patients less than 70 years of age, and the Alliance study is frontline for patients over 70 years of age, and I'll get into some of the details on the differences in these studies. You want to go to the next slide. So really focusing on the S1925 EVOLVE study, which is a randomized phase 3 study of early intervention with venetoclax and obinutuzumab versus delayed intervention with the same regimen in these newly diagnosed asymptomatic high-risk patients with CLL. I've listed out several members of the SWOG committee below that have helped in the development of the study, and our ECOG and Alliance champions here if you have any additional questions. You can go to the next slide. Asymptomatic CLL patients actually in 2021 actually hasn't changed since 2020, or hasn't changed for many years, as the current recommendation is watch and wait. And this is mainly due to the fact that prior interventional studies with agents such as standard chemotherapy agents, chlorambucil, fludarabine, really failed to show an overall survival benefit. We have a lot of great prognostic tools right now, and one of the ones we're utilizing in this upcoming study is the CLL International Prognostic Index, because it has been a great predictor of survival. The factors included in this are the presence of deletion 17p or TP53 mutation, which is a very strong predictive factor. As you see, it's given 4 points in this scale. Also, serum beta 2 microglobulin, the IGHV status, rise stage, and age. When you put these factors together, a score of 4 or higher is considered high, and the 5-year overall survival of these patients from diagnosis is only 53%. And in the very high-risk population, it's only 19%. Less than 50% of these patients in the high or very high-risk group are treatment-free at 5 years from diagnosis. So this is really a group of interest for early intervention. Go to the next slide. And as you all know, that the frontline chemoimmunotherapy has really been mostly replaced by targeted therapy. And for this particular study, we have focused on the combination of venetoclax and obinutuzumab because these is a time-limited therapy that can induce deep MRD undetectable remissions. This is a time-limited therapy that was studied in a phase 3 study, the CLL14 study, as compared to chlorambucil and obinutuzumab. And it's a limited of 1 year of venetoclax therapy. 76% of patients were able to achieve MRD undetectable remissions at 3 months following this therapy. So this regimen of venetoclax and obinutuzumab is effective, tolerable, and time-limited, and is really an optimal regimen for early intervention. And the question we're asking in our SWOG study, the S1925 study, is can early intervention with venetoclax and obinutuzumab prolong the survival in these high-risk asymptomatic CLL patients? Next slide. Here I've highlighted the general schema of the study. Again, this is for newly diagnosed patients with CLL who are asymptomatic, have a CLL IPI score of 4 or higher, and or have complex cytogenetics, which is defined as 3 or more genetic abnormalities on stimulated karyotype. These patients are enrolled and stratified high risk versus very high risk and randomized 2-to-1 to early versus delayed therapy with this venetoclax and obinutuzumab regimen. Again, this regimen, the obinutuzumab, lasts for the first 6 months. The venetoclax starts at the beginning at the 3rd week of therapy and lasts through 12 months of therapy. The primary endpoint of this study is a high bar. It is overall survival. And in order to achieve that primary endpoint goal of determining whether early intervention can improve overall survival, we need 247 patients. We do have multiple secondary endpoints planned, including safety, overall response rate, duration of response, progression-free survival, time to next treatment, MRD status, and quality of life objectives. We also have multiple translational endpoints planned around MRD status and if there's any patients who develop resistance to therapy. You can go to the next slide. I just want to highlight the inclusion criteria for this study. These patients need to be diagnosed within 12 months of enrolling in the study. They must not require therapy by IWCLL guidelines, which means they must be asymptomatic with no cytopenias. They must have no prior CLL-directed therapy. Again, CLL IPI score of 4 or higher or complex cytogenetics. ECOG 0 to 2, and no issues with GI malabsorption as venetoclax is an oral therapy. And go to the next slide. The status of the study, we're very excited to have activated the study in December of last year. We are working on a planned amendment because as sites come online, we just want to clarify a few items that are in the protocol. And one thing that I specifically want to point out to the group is that there's an error that looks like patients need a bone marrow biopsy at registration, but they actually don't. They only need a bone marrow pre-treatment. So if they get randomized to the delayed arm, they don't need to have a bone marrow right up front. There is a memo that accompanies the protocol that states this, but I just want the treating physicians to be aware of that so nobody gets an unnecessary bone marrow biopsy. There have been quite a few sites come online just recently. I think sites are really just getting this open, but I just listed a few that are open. We have been able to accrue a few patients here at University of Utah. I think one really important thing when you discuss this study with your patients is that all of these drugs are provided free of charge for both arms. And so whether the patient gets early intervention or delayed intervention, these drugs are still paid for, which I think has been very exciting to a lot of the patients that I've spoken with. Also to note, clearly, COVID needs to be taken into consideration during this time. And we do have a pretty generous period of time between enrollment and actually starting on early intervention. If the patient hasn't been vaccinated for COVID yet and they want to get vaccinated before there's a chance that they might get randomized to early intervention, there's about a month between screening and randomization. And then there's actually another month between randomization and when the patient would need to start therapy if they were to go on early intervention. And so if the patient is really concerned about making sure they get in their full series of COVID vaccines before they could potentially start therapy, the protocol does allow for that. And so if you want to go on to the next slide. I just want to show how this fits in again with the other cooperative group studies. So our study is for all of these studies that are currently ongoing are for frontline patients, but our study is for all ages, only high risk and asymptomatic diagnosed within the last year. The ECOG and Alliance study, I'm going to talk about those on the upcoming slides. But again, the ECOG study is for patients less than 70 years of age and is almost completely accrued. And I'll give you more details about that. The Alliance study is for patients older than 70 years. I'm going to go to the next slide. So the ECOG study, again, this is a frontline study, and it does randomize patients to ibrutinib obinutuzumab versus ibrutinib obinutuzumab and chlorambucil, and this is for younger patients. The study chair is Tate Schonefeld from Stanford, and our SWOG champion is Dr. Shadman at Fred Hutch. You go on to the next slide. This really highlights the schema for this study. Key eligibility are CLL or SLL patients who have an indication for treatment. They cannot have deletion 17p, and they must be younger than age 70 in this study. Again, this does compare a 2-drug regimen of ibrutinib and obinutuzumab versus a 3-drug regimen of ibrutinib, obinutuzumab, and venetoclax. In the 3-drug arm, all study treatment does complete at the end of cycle 19. In the 2-drug arm, ibrutinib is indefinite therapy. Again, all the study drugs are paid for on this study, and the primary endpoint is progression-free survival. And go to the next slide. So really up-to-date news on this study. The primary accrual goal has been met. There's planned 720 patients. When I look at the website, they actually say that 734 patients have been accrued. And so the study really has a planned closure date of just in a couple of weeks in April, the end of April this month. And so you know, if you were planning to get your patient on this study, there are likely no spots. And before you start any screening procedures, please reach out and make sure that that is allowed to screen those patients, because this really, this study has really accrued very, very quickly. You go to the next slide. So a study that is still open and ongoing is the Alliance study, and it's a similar study in that it is studying a 2-drug regimen of ibrutinib obinutuzumab versus a 3-drug regimen of ibrutinib obinutuzumab venetoclax, and this is specifically designed to be for treatment-naive patients who are 70 years of age or older. The main study chair is Jen Woyak from Ohio State, and the SWOG champion is Brian Hill from Cleveland Clinic. Go to the next slide. The primary objective of the study, again, is progression-free survival. Secondary objectives include looking at an MRD undetectable CR rates, overall survival, and the toxicity of the regimens. You go on to the next slide. Here is the study schema. Again, this is for previously untreated patients who are 70 years of age or older. This study does allow patients with 17P, which is different than the ECOG study who excluded those patients. Again, these patients are randomized to two-drug therapy with ibrutinib plus obinutuzumab versus ibrutinib plus obinutuzumab plus venetoclax. We go to the next slide. All of these patients at the end of cycle 14 or beginning of cycle 15 are going to have an MRD kind of formal response assessment. In the true drug arm, whether they are MRD positive or negative, the plan is to continue ibrutinib therapy. In the 3-drug arm, if they have an MRD negative CR, they will be discontinuing ibrutinib. But if they have any response less than that, they will continue ibrutinib indefinitely. Again, all of these very expensive drugs are paid for by this study. And so I think it's a really great benefit to patients to go on this. You go to the next slide. Again, highlighting the eligibility criteria. This is for patients 70 years of age or older, ECOG 0 to 2. Neutrophils need to be over 1,000, platelets greater than 30, except in the case of bone marrow involvement, which I never understand that because every patient with CLL does have bone marrow involvement. Creatinine clearance is greater than 40 in normal liver function tests. You go to the next slide. So for this study, the number already enrolled is 283 as of yesterday out of a planned 454, so a little over halfway done here. And so there is a planned amendment under review. None of this is finalized yet and still has to go through CTEP and all the appropriate pathways, but there is some thought to a future amendment allowing patients 65 and older to go on this study, as the ECOG study accrued very quickly and is close to enrollment. But again, that amendment has not been passed yet. Also, notably, this study does currently exclude SLL patients, but there is a planned amendment to change that and include SLL patients. Again, that has not completed the formalized CTEP review, and so that may change over time. If you want to go on to the next slide, and you can click through again, just to remind you, our SWOG study, frontline, asymptomatic, high-risk patients, ECOG Akron has planned closure in a couple of weeks here, and the Alliance study is ongoing for patients 70 years of age or older, regardless of 17P status. That's the end of my presentation, but happy to take any questions.
Thanks, Deb. Very exciting time in CLL, as it has been for the last decade. So I just want to emphasize the opportunities here. I think tomorrow's the last day actually to consent your patient for the ECOG study, and next Friday is the last day to register them. So we're reconvening a CLL working group among the 3 cooperative groups, and I invite anyone who's interested to contact Deb or myself. We really are interested in ideas for what we should do next, in particular for upfront CLL. Let's see, I don't see any specific questions. So Deb, thanks again for leading that study. And I think we'll move on to our next major topic, which is acute myeloid leukemia. Harry, you want to give a start us off?
Thanks, Steve. Yeah, I will give an introduction to what we're doing in the myeloid neoplasms, AML, and MDS. Frank, I'm not sure if you would, can I have control of the slides or?
Sure, stand by 1 minute.
Okay. So we have had a number of different names for what we've been working on for the past few years, but this is the final name, MyeloMatch. And quite frankly, it was proposed by Jim Doroshow himself, and it makes a lot of sense because, as you will see, much of the diagnostic evaluations that will be incorporated into our initiative are being shared with other match programs, and also the informatics will be shared. And so his idea was to maintain the match part of it, which actually, if you didn't know, stands for Molecular Analysis for Therapy Choice as a precision medicine trial. So as opposed to combo match or immuno match, this is Milo match. So let's start. Let's get rid of that. Okay, so what are the aims of Milo match? And I actually like to see this, and it's not on this slide, as a response to the 2010 Institute of Medicine report on the future of the cooperative groups. And there was a very important summary statement in there where the Institute of Medicine said basically the cooperative groups fulfill a very important function. However, if we are going to be successful in improving the outcomes of Americans with cancer going into the future, we are going to need to develop pathways and algorithms that in protocol development that involve every all of the partners that are involved in that. So industry partners, government, regulatory, FDA, the academic groups, patient advocates, and industry, where many of the agents are coming from. And so the goal of Milo Match with that imperative from the Institute of Medicine is to build a portfolio of clinical trials that will help accelerate current and future precision medicine initiatives, culminating in the advancement of potential therapies for myeloid malignancies. And to do this, we're instituting what we hope will be seen as an efficient operational model that will be attractive to our industry partners and to the NCTN sites. And what we're designing are multiple unique studies that will be done in parallel, as you will see. And the studies will allow for investigational drug combinations across industry partners. Okay, so by doing this through CRADAs that are developed with the NCI, we are hoping that we will be in one of the best positions to do novel drug combinations, not just with novel drug combinations that belong to one industry partner, but in the best in class from different industry partners. And as Steve alluded to before, a driving force in everything that we've been doing in the intergroup in developing studies is to do what our predecessors did decades ago, and that was to develop the careers of young investigators. In fact, many of the senior people who have moved on made their early careers as young investigators in cooperative group studies, and we'd like to bring that back and ask them and promote them for leadership positions throughout the clinical trial portfolio, as well as the laboratory program. Oops, let me go back. Okay, there was a bit of a delay. And in general, we are going to try to leverage the advancements in next-gen sequencing, that being duplex next-gen sequencing or single-cell mutational profile, leverage that technology to try to impact on the outcomes of patients with AML along their disease pathway. So the principles that we have laid out to investigators interested in joining us in this initiative is that the subjects will only enter the MyeloMatch initiative at their initial diagnosis prior to therapy. So no one's going to be grandfathered into this. We are going to look at the natural history and the response to various therapies in patients from the time of diagnosis. The importance of that is that we collect genotypic data from the time of diagnosis, as well as important information about the physical characteristics of those patients in some of these trials, such as for older patients, frailty indices, and geriatric assessments. The protocols are going to be designed to identify novel therapies that are worthy of further definitive testing in phase 3 studies. So this is another message that we've gotten out to our investigators. We're not looking for the next large phase 3 study that'll take a decade to complete, such as Ratify, which has clearly been important in changing therapy for AML, but took a long time. What we are doing here is we want to identify things that are worthy of that kind of phase 3 development. So what MyeloMatch focuses on are smaller, right now mostly randomized phase 2 studies to identify novel therapies that result in a meaningful improvement in some early response assessment. And as I said, in leveraging the breakthroughs, advancements in next-gen sequencing, many of these studies are looking at MRD negative remissions. And importantly, is that if patients are registered at the beginning, they will be followed through all of the clinical trials in the different tiers of MyeloMatch, which I will define. So what is this operational efficiency that we've been working on? And here you see it. We've developed a number of committees with collaboration between the various cooperative groups, and very importantly for this process, the NCI itself at a very early stage. So in the Senior Scientific Council that is constantly deciding the direction of our scientific advancement, we are working directly not only with the academic leaders of the different cooperative groups in Canada, as well as in the United States, but also with the many different divisions of the NCI that are important in clinical trial development. So we're all at the table. So again, hearkening back to what the Institute of Medicine challenged us to do. And the Senior Scientific Council meets every week. It's a 1.5-hour meeting every week. It's been going on for well over a year. You know, we keep minutes and agenda, and you'll see what we've been working on. Now, we work closely with two other committees, an Agents and Genes Committee and our Laboratory Committee, which I will show you in more detail. This more senior group of people relies on the hard work of the people with their boots out on the ground, the clinical basket working groups. And right now we have 3, the myelodysplastic syndrome group, older AML, and younger AML. And at some point in the future, we may be adding myeloproliferative neoplasms to this portfolio, but not quite yet. I and Mark Litzow have been asked to lead the Senior Scientific Council, which we do. Agents and Genes Committee is led by Sarit Asuline from the Canadian Cancer Trials Group, Celine Luger from ECOG Akron, and Rich Stone from Alliance. The Laboratory Committee, John Bird, who is moving from Ohio State to be the Chair of Medicine at University of Cincinnati. Congratulations to him. Chris Horrigan, Jerry Radich, and by the way, parenthetically, I really hope John Bird remains involved in this process. He's been instrumental in helping us develop things, and from his vast experience in Beat AML, very important input into the design of MyeloMatch and how we can work together. All of those names, by the way, are in alphabetical order. Now, the clinical baskets are groups of investigators that will work together to develop clinical trials that you're going to see. Younger AML, so these are intergroup collaborations, but they each have to be led by somebody. And so we've spread it around. Ihab Atala, University of Wisconsin from ECOG, leads the younger AML working group. Older AML is led by our own Laura Michaels and Jeff Wee, and they take a slightly different approaches to this. Jeff Wee, for the older patients, may be fit for chemotherapy, however we define that, and Laura Michaels for the older unfit. And then Toyosi Odeniki from University of Chicago Alliance leading the MDS group. So this is what we envision. At the time of diagnosis, the patient will go into a master screening and reassessment protocol. I'll show you some of the assessments they do based on their physical characteristics and genomics, cytogenetics. They will be allocated to a initial therapy, so-called induction therapy. From there, they may go on to a post-remission trial, specifically looking at understanding clonal selection and treating patients based on measurable residual disease. Tier 3 will be other consolidation and transplant studies, and we are working closely with the BMT CTN. Our own Pat Stiff has been approached. I'm not putting you on the spot, Pat, but he's been approached to lead the BMT CTN, or the transplant group of MyeloMatch, and I hope we can entice him to do that. And then a tier 4, where we're looking at really deep sequencing of samples to see if we can intervene at an early time point against specific mutations. So you're going to see some of these clinical trials, but being discussed by Paul in this younger AML working group from this younger AML basket working group. And also you'll hear Dave Solomon talking about an older AML study. So I'm not going to go into details here, but suffice it to say that the way we're going to make this work is that in MyeloMatch, each of these protocols will belong to the various investigators in various cooperative groups. So everyone stays involved in every section of myeloid neoplasms, AML, MDS, younger, older. And we look to the leadership of our working group basket leaders to assign studies to the different cooperative groups, so that each of our operations offices and statistical committees remain engaged across the spectrum of myeloid neoplasms. We've been pretty busy. These are agents of interest for MyeloMatch. I won't go through the whole list there. Some already FDA approved, others in development that leukemia doctors think are highly promising. We have already had meetings. As I said, every Friday we meet discussing 13 agents. 4 CRADAs are, this is as of January, 4 CRADAs are already in place for MyeloMatch. 1 CRADA is in process, and many additional agents are under discussion that are not listed here, but part of our ongoing weekly discussions. Now, how are we going to make this happen? As I said, the NCI is going to provide the support of the molecular assays for these 3 new precision medicine initiatives, MyeloMatch being one of them. The network of harmonized laboratories is led by the scientists at the Frederick that are listed there, plus the MyeloMatch Laboratory Coordinating Committee with our laboratory group leaders and Lindsay Harris from the NCI. The MyeloMatch Master Screening and Reassessment Protocol, at the time of patient entry and at subsequent reevaluations as patients move from tier to tier, will employ various assays. We cannot tell you more details yet because they are still being negotiated and contracts finalized, but we are going to provide rapid turnaround of molecular and cytogenetic information to inform treatment decisions. And you can see the NGS platform that we will be using. And finally, as we collect these samples on patients, they will be distributed, they will be banked, and some of the samples may go to the cooperative group leading a study for embedded clinical laboratory assays that they have interest in and are embedded in the protocol. But it's quite an undertaking. We've already had protocols going to the Leukemia Steering Committee with feedback, and we are very hopeful to see the first protocols of MyeloMatch activated by the end of this year. Steve, I'm sure I took more than my share. I apologize, but I'll turn it back to you.
Thanks, Harry. That was a great overview. You know, again, I think it's going to be a really, really exciting time in myeloid neoplasms. This is the role that the cooperative groups were built for. And so hopefully we'll continue to make progress in this common disease for us. I'm going to turn it over next to Laura Michaelis, who, as Harry mentioned, is the leader of one of the aspects of the MyeloMatch program, and Laura will guide the rest of the AML presentations with her colleagues. Frank, it might be easy to turn the presentation over to her so she can advance slides.
Great, thank you so much. So as Harry mentioned, this is the overall sort of bird's eye view of where we stand, and I put a circle here to show that SWOG and we've played a role in all of this, but we want to focus a little bit on the working group that's for older patients. You can see here that within each basket, if we think about the older, fit and less fit patients, we're hoping to have multiple protocols that are targeted for patients within those baskets and that would not overlap necessarily with patients or with protocols from the MDS or younger basket. Next slide. Well, actually, I can do that. Hold on. So as Harry mentioned, Jeff, we and I are leading these older working group, and our aims are to investigate the role of the targeted therapies, validate flow cytometric based minimal residual disease, provide a platform that would validate these new laboratory assays, and develop core geriatric assessment and quality of life measures that would inform the interpretation of the trial results and characterize treatment benefit. And part of that, I'm going to draw your attention when we see Paul's presentation, part of that is also going to be the ability to randomize people to less intensive and more intensive regimens despite fitness. And that's also an important part of us learning whether or not we can, whether or not we need to limit the intensity of therapy based on fitness. And finally, we want to engage and provide mentorship to early and mid-career investigators, as has been mentioned. So this is the this is the younger clinical basket algorithm to date. Harry just mentioned that. And I want to point out that Paul Shammy is going to talk in a little bit about one of the protocols that's been led by SWOG here, and this is the high risk protocol. He'll go over where that is and where we stand. But this is for younger patients. And in the younger patient, the 3 trials that are furthest along is one that will be an MRD-driven phase 2 study of venetoclax for newly diagnosed intermediate risk patients, one that's an MRD-driven phase 2 study of venetoclax plus chemotherapy for those with high-risk AML, again by ELN criteria, and finally one that's for designed to eradicate minimal residual disease in patients who achieve CR but where there's still evidence of disease. In the older clinical basket, we've divided ours into those who are fit, and we're looking at if we already have a FLT3 wild type trial that's ongoing in patients who are eligible for intensive chemotherapy. That trial predated the development of MyeloMatch, but that's run by Jeff Wee, and that's already open in many centers through the Alliance. We also are looking at a fitness independent trial, which will be for P53 mutated disease. David Salmon's going to talk about that in a second. And we've developed trials for IDH mutated, 1 mutated, IDH 2 mutated, and FLT3 mutated patients who are in the less FLT. So if we think about how this divides up, we have a SWOG-led study here that'll be P53 mutated. We have an Alliance-led study for IDH1 mutated, and IDH2 mutated is also SWOG-led by Eric Husselton. We're going to hear about that in a second. Again, these are for older patients who are over the age of 65. We're also looking at a FLT3 mutated study and one that for patients who don't have a targetable marker, which is being led by ECOG. All right, so I want to give the podium now to David Salmon. He's going to talk about a proposal that we have for P53 mutated AML. David?
Sure, can you hear me okay?
Absolutely. I can advance if you want. I'm already in control.
Perfect, yeah, I am slightly scatterbrained. I'm inpatient and then there's just other call at the same time, but happy to really present here. Very excited to to move this concept forward. I've been working very close with Rory and Amir, on this protocol. So they, have actually come up with this very fancy name, or eradicate TP53 mutated AML. I do not hold any intellectual property in the name of this trial. Next slide. This is just a really kind of, an overview that everybody knows, but of course, P53 population-wise is probably more in the 8 to 10% realm. But if you go to the next slide, I think in many of our cohorts, for example, at our center, actually P53 AML represents about 20% of all our patients, probably secondary to referral bias, sickness, and sort of the increasing recognition that this group really does quite poorly. Next slide. And this is just one prognostic curve. I think we can go into detail, across studies, but really the median overall survival in almost all P53 mutant studies to date has been close to 6 to 7 months. Is probably what the outcomes are, really independent of therapy, and that's including patients that get response. And I think, as we see on subsequent slides, response, particularly for this patient population, is not a good prerequisite for how they ultimately do, as really the durability of the responses is most critical. So you could see responses, including with more newer therapies such as CPX-351, HMA venetoclax, with different schedules. You get varying, CR rates. Personal experience has been lower with intensive chemotherapy, but again, I think despite this, the survivals on these trials are very poor. Next slide. And so this is just looking, again, at why, despite a response rate with HMA venetoclax of 50%. You still, in earlier, phase 1b/2, median OS was 7.2 months. In the phase 3, there was no clear benefit. And then there's been several publications at ASH of this past year, again, looking even at extended schedules of HMA, and the median OS was really 5 to 6 months across those groups for P53. So, of course, looking at novel therapies for this patient group is what we're all excited to do. We presented data with APR with azacitidine. Although the we only allowed oligoblastic AML, at least in the U.S. study. There were some patients with more overt AML in the GFM study led by Clouseau and colleagues. But we again see high response rates, median OS starting to be potentially somewhat improved, although small numbers. And then with magrolimab azacitidine, these data were most recently presented at ASH. The follow-up was short, but a very high complete remission rate, and many of these remissions continue to improve over time. And although follow-up is short, median OS was 12.9 months, but may actually be significantly extended. And I think we look forward to later data cutoffs on this. Next slide. So I think what's emerging with CD47, and this is not just specific to magrolimab, but really across CD47 programs, is that really the balance of pro-AE signals is likely the key, probably one of the key mechanisms for synergy. So agents that may upregulate pro-AE signals may ultimately induce synergy. And so this is just showing some preclinical work that was done with Apria, where APR-246 can actually robustly lead to upregulation of calreticulin, actually higher than azacitidine, which several groups have shown can increase the pro-AE signal, and has really been the basis for the initial combination of those two drugs, but really giving some rationale why APR may be a good agent to combine with CD47, especially given high response rates with both of these novel agents. Next slide. I would say, we have a slide on it, but similar data was at least shown by looking with ALX048 with venetoclax. So I think actually all three drugs now have been shown to increase pro-AE signals, just giving a little bit more of the rationale for the study. Of course, looking at oral options, for patients improves, adds some level of novelty to it. Of course, the agent ASTX727, is approved in MDS and CMML patients, and thus, is being combined similarly when HMA is indicated in AML patients. So next slide. So really, this is the most important. So this is really our concept schema. So the control arm is ASTX727 and venetoclax, really at a standard schedule. but does allow for an all-oral option to give sort of some novelty to the control arm. And then we have 3 different combinations, including HMA venetoclax and a CD47 target therapy. Potentially lead right now is TTI-621, but there are several others that are potential, and we can talk about that, as well as a triplet with APR-246 and venetoclax. I would say both triplets are ongoing in investigator-initiated or sponsored studies in smaller settings. And then quite interested in sort of a non-venetoclax-based triplet arm of ASTX727, TTI-621, and APR-246. And if we go to the next slide, I think it gets into the some of the statistical design. We're going to do this as fitness independent, so really just age over 60, but could be fit or non-fit. And really, given the poor correlation of response rates and overall survival, the design of the study is powered to look for an improvement of OS as the primary endpoint. Each arm will be individually compared to the control arm. There's not cross comparison. You can see the statistical thresholds, regarding the power and alpha, and there will be an interim analysis for futility, which may close any of the individual arms if they may not look like they will ultimately reach the primary endpoint. Next slide. So just for some key, technically these are new combinations with ASTX727. So there will just be a very brief 6-patient lead-in, although we really anticipate no issues from that perspective. Again, 2 out of the 3 triplets have already, ongoing in phase 1 study and have been safe. There will be a stratification based on P53, given we and others have shown, greater or less than 40% to be a strong prediction factor for outcomes. And I think there are some important considerations. There is a new 2nd-generation APR agent, which we're opening quite soon, that could be, which is oral versus IV, and potentially could be implemented, but likely more with APR-246. And then again, whether or not TTI-621 ends up going forward, lenzulamab, magrolimab, ALEX-148, these are options given may not be specific to any one agent, and we're of course excited to try to get this trial open in the near future.
David, thank you so much. Steve, do you want to take questions at the end?
Yes. Why don't
Okay.
you do that?
So David, if you have to step away back to rounding, I can forward on those questions from the askers.
Sure, happy to do that. Thank
or
you
you
very
can
much,
stay
Laura.
on. That would be great too. So next
Okay.
I want to introduce Eric Husselton, who's leading our trial that will be open for patients with IDH2 mutated AML. Eric, I can forward for you.
Thank you, Laura. So our trial is a randomized phase 2 trial of the oral decitabine compound CDAC or ASTX727 combined with venetoclax and enasidenib compared with azacitidine and venetoclax for patients with IDH2 mutant AML. Next slide. So just briefly some background. We know that older adults with IDH2 mutant AML do well with azacitidine and venetoclax. And in the phase 3 study published in the New England Journal comparing aza-ven versus aza alone, the overall response rate was 75% for the IDH2 mutant patients compared with 66% overall. Likewise, the 1-year overall survival was also improved at 67% for the mutated patients versus 55% overall. And it's been estimated that the rates of MRD negative remissions as measured by flow cytometry are higher at about 40% with mutated patients versus 30% overall. However, despite these good clinical outcomes, as you can see in the figure in the bottom left, patients continue to have molecular MRD. And these are variant allele frequencies showing about half of patients in remission continue to have very high levels of IDH2 mutations, and almost all the patients except for one continue to have some detectable mutation. Next slide.
Sorry.
So just some rationale for our study. We know that enasidenib is an oral IDH2 inhibitor that's active as a single agent and in combination with azacitidine in the upfront setting. On the right, it's a figure from ASCO last year comparing the combination of enasidenib with azacitidine compared to azacitidine, showing improvement in event-free survival. And most interestingly, it showed a much higher level of IDH2 mutation variant allele frequency suppression compared to azacitidine alone. With the combination causing a reduction about a median of 70% compared to 14%. And then there's PDX models of human IDH2 mutant AML that shows the best responses were seen with concurrent treatment with IDH2 inhibition with venetoclax compared to either agent by itself or sequential therapy combinations. And currently there's an ongoing trial of the IDH1 inhibitor, ivosidenib, venetoclax, and azacitidine that showed a very high response rate in the upfront setting that was presented at ASCO last year. So we hypothesized that adding enasidenib to the hypomethylating agent venetoclax combination would improve the rate of complete remissions without measurable residual disease. Next slide. So this is our trial schema here. The key eligibility would be patients over the age of 60 with IDH2 mutation who are unfit for standard chemotherapy. They'd be randomized 1-to-1 to receive the standard of care, venetoclax and azacitidine, or to a novel all-oral treatment regimen of venetoclax, the oral decitabine compound CDAC, and enasidenib. Responses would be assessed with a marrow biopsy after cycle 2, in which case patients would then go on to receive maintenance therapy with continuous cycles until disease progression or intolerability. Next slide. So some key endpoints here. Our primary endpoint, again, is the rate of MRD negative CR with or without count recovery as measured by multi-parameter flow cytometry after 2 cycles of therapy. And key secondary endpoints are looking at MRD as measured by molecular and flow cytometry in both the blood and the marrow after cycles 1, 2, and 4, and then every 2 months in the blood and 6 months in the marrow, with detectable MRD in the blood prompting an early marrow biopsy. We would be looking at relapse and event-free survival, response rates, rates of transfusion independence, and overall survival, as well as toxicity, and perhaps most importantly, the association between flow and molecular MRD and clinical responses. Next slide. So our null hypothesis is the rate of an MRD negative CR with the control arm aza-ven is about 40%. And we hypothesize that this triplet regimen will improve the rate of MRD negative CRs to about 65%. This will require about 88 patients randomized 1-to-1 to give us an 83% power. And we'll do an interim analysis after 44 patients with early termination if the triplet response is not better than the standard of care. Just to review study progress, we worked on this with our older adult basket working group and presented this to the Senior Scientific Council in September, and then with help with Chris Corrigan at the NIH, did some additional revision cycles and have got this approved by the Senior Scientific Council this spring and are in the process of submitting to CTEP. So that, thank you.
Thank you so much, Eric. I want to do a couple of call outs. First off, both Eric and David really have been helped along the way by Megan Othis and our statistical team. So I want to thank so much. Megan's she's like a hero helping out with this. And I also want to thank the Senior Scientific Committee, which is assigned kind of liaisons to coach the groups as to how to tweak this around. And I also want to point out that both of these studies are powered with not a huge number of patients, but with real meaningful endpoints. So if we can keep these numbers nimble and small studies that can open and close and move forward, that's one of the main goals of MyeloMatch, is to continue having trajectory forward and real traction that way. So I want to next go. So this is just an overview of some other studies. We're cloning Eric's study for IDH1 mutated patients. We've talked about the P53. We also have some studies that will be led for P53 mutated AML, and we're looking again for those study for HMA refractory patients. So next, I just want to introduce Paul Shammy, who's going to talk about the SWOG-led studies for younger patients with high-risk AML. Paul, I'll be happy to advance for you.
Okay, thanks, Laura. So this study is part of the MyeloMatch program, and I'm not going to go over MyeloMatch again. Obviously, we've had a great overview by Harry. So that's a randomized phase 2 study of novel therapies for younger patients with high-risk AML. The primary objective is to compare measurable residual disease negative CR between each of the experimental regimens as compared to a 7+3 control. Secondary objectives is to look at toxicity of each of the regimens, and then to look at the different remission outcomes, CR, CRI, and then event-free survival, relapse-free survival, and overall survival. Then another objective would be to try to use the molecular information to see if we could find any mechanistic evidence of specific resistance. Next slide, please. So the inclusion criteria are pretty standard: newly diagnosed AML, non-APL. Patients need to have high risk per ELL criteria, but also ELN criteria, but also patients with antecedent hematologic disorder and therapy-related AML would be eligible. Obviously, we're excluding FLT3 mutated AML, which is a separate trial, and then the rare cases of a 922 mutated AML. Younger patients, 18 to 59, reasonably adequate performance status up to 3, and normal or acceptable ejection fraction. Next, please. And then normal organ function, we're allowing prior malignancy, pretty standard, as long as they're not requiring concurrent therapy. And then HIV hepatitis is allowed according to the ASCO FOCR criteria, obviously the usual contraception requirement. Next slide, please. Exclusion criteria. So these are newly diagnosed patients, so no prior therapy for AML except for Hydrea to control the white count, no concurrent investigational agent. Kind of can see why people would be getting CPX-351, but if anything that's similar where they could be allergic to, then anything that would be uncontrolled intercurrent illness or any psychosocial issues that would prevent being enrolled. That's the general schema. So this would be a 4-arm 1:1:1:1 randomization with a standard 7+3 as a control. The second arm would be cytarabine and daunorubicin along with venetoclax using the regimen that is currently under investigation at Dana-Farber, led by Rich Stone. We already have, based on Rich's ASH presentation, we already know the safe dose of venetoclax to combine with 7+3. And then the other two regimens are CPX-351 along the standard induction, and then AZA venetoclax likewise with the standard induction regimens. So all these regimens are known, have been published, except for the 7+3 venetoclax, which is in development, but has already been presented in a preliminary fashion. The statistics are, as we can see. I hope Megan will save me from any embarrassment if I'm making a fool of myself, but this is a 1:1 randomization for each arm. There will be stratification along ages, 18 to 39, and then 40 and older, and stratification along performance status. 60 patients per arm. The null hypothesis is MRD negative CR rate of 35%, which is what was observed in the SWOG 0106 trial with the 7+3 regimen. The alternative hypothesis would be a 20% absolute improvement with an MRD negative rate of 55%. There will be an interim analysis after we have 30 patients enrolled on each arm. Next, please. And then the final analysis would be a 1:1 comparison of each of the experimental regimens as compared to 7+3. The EFS, RFS, and overall survival will be estimated using, standard Kaplan-Meier method, and then toxicity will be evaluated as shown. Next slide, please. Now, where do we stand? The concept has been generated and approved at Triage. We have an initial, I would say, very rough draft of the protocol. It's a work in progress. The concept has been reviewed, I want to say 2 or 3 weeks ago, within the past months, by the Leukemia Steering Committee, and we got the feedback of the committee, and we are working as we speak to, revise the concept, basically answer the questions that the steering committee has asked, clarifications, etc. So this is, again, in the process of being revised. We should have that resubmitted to the steering committee, hopefully within the next 7 to 10 days. And we're hoping to open this maybe by the end of the year. And that's the end of my presentation. Thank you.
Thanks so much, Dr. Shami. So I will turn this back over to Steve and Harry for questions about, but I just want to compliment, these are all 3 SWOG-led studies, which are foundational for this Milo match to work. And I really appreciate the work that all our presenters have put into developing these. So exciting times. Thank you.
Thanks, Laura, everyone. Yeah, I agree. Very exciting for AML right now, and you can see things are working their way through the system. I noticed that, for example, one of the erase protocols will be considered at the Leukemia Steering Committee next week as well. So stay tuned. Parenthetically, I know acute promyelocytic leukemia is not part of MyeloMatch, right, Harry? Is that correct? But it's
that is correct.
Certainly near and dear to my heart. And there will be a protocol coming through ECOG for oral arsenic, believe it or not. Oral arsenic, atra, and gemtuzumab for high-risk APL. So we look forward to that. And for any young investigators who would like to be the SWOG champion, please reach out to Harry or Laura, and we'll see what happens. So let's turn next to our other major acute leukemia, acute lymphoblastic leukemia. We've got a really dynamic working group that's been very productive, led by Wendy Stock of the Alliance, with a representation here you can see across the cooperative groups, and in particular, Kristen Dwyer, Angela Advani, Ali Jabor, have been instrumental in developing some of these protocols, as well as several other investigators within SWOG that you'll hear about. So I'll turn this over to Kristen.
So this is Anjali. I think Kristen and I were going to split this up. So I think I was going to do the first part and Kristen was going to do the second part. Correct, Kristen?
Okay, perfect. Take it away.
Yeah, go ahead, Anjali.
So as Steve and Kristen both mentioned, this has really been a great collaboration among many investigators and groups. And also I wanted to thank Kristen for putting together these slides. So first we're going to talk about an update on some of the closed trials, some of the ongoing trials. But for most of this, I'm going to actually have Kristen. Kristen is going to actually talk about the upcoming trials, which I think are really the most exciting part. And so both she and Eli Jabor will talk about those. So very briefly, the first trial I'm going to talk about is S1318. This is a trial for elderly patients with ALL. This trial had 2 portions, a pH negative cohort and a pH positive cohort. Kristen O'Dwyer was the co-chair of this study with me. And so for the pH negative cohort, the manuscript is actually drafted, and so hopefully we should be submitting this soon. In the pH negative cohort, this was a trial where we used blinatumomab for induction and post-remission treatment, followed by POMP maintenance. The complete remission rate was 66%, so quite encouraging, with a very high rate of complete molecular remissions. And most importantly, at 3 years, the overall survival is 37%. So quite encouraging, given that this was really a single agent immunotherapy in an older patient population.
For the pH positive cohort, this cohort just recently completed accrual, and so this data we need to put together and are analyzing this. So next, I'm going to talk about, we can go to the next slide. For the next slide. Oh, perfect. So we're going to next talk about some of the other ongoing trials. Next slide. So this is A041501. This is a trial for young adults with newly diagnosed B-cell ALL. So this is a U.S. intergroup trial, but being led by Dan D'Angelo from the Alliance. So this is for patients that are CD22 positive B-ALL, patients less than 40 years of age. They receive an induction, which is C10403 induction. Those patients that are CD20 positive receive rituximab. And then the randomization is for patients to either receive 2 cycles of inotuzumab or no inotuzumab, followed by standard consolidation and then maintenance. This trial has a planned number of patients of 341. So far, 179 patients have accrued. There's currently an amendment approved to add blinatumomab for patients that are MRD positive following course 3. And there's been discussion, we're going to talk about later about Eli and Marlisa's trial in elderly ALL. But there's been discussion about potentially increasing the age of this trial to 40 to 50 years to kind of fill that gap as the elderly ALL trial opens. And if we did this, we would have PEG dose adjustment. Next slide. The other trial that is currently ongoing that's being led by Matt Wiedewald, also of the Alliance, is A041703. This is a trial sequencing inotuzumab and blinatumomab for pH negative CD22 positive B-ALL. This trial has 2 arms. One is a newly diagnosed elderly arm, and then the second arm is for patients with relapsed or refractory disease. So this trial, as mentioned, is ongoing. There are 31 patients accrued to date. Next slide. So the other trial that's recently, fairly recently opened has been EA9181. This is a trial for newly diagnosed patients with pH positive B-ALL. This trial is being led by Yishai Ofren from ECOG, but Katie Phelan from SWOG, who's from Loyola, is our SWOG lead for this trial and has done a really fantastic job. So for this study, all patients will get TKI and steroids as the run-in. The TKI can either be dasatinib or ponatinib per physician's choice, and this was because there was a lot of debate about which TKI to use in the upfront setting. The patients, the main question is kind of the induction question. So patients get either TKI and steroids plus blinatumomab versus TKI and steroids plus chemotherapy. Patients are then have MRD testing done. And we can go on to the next slide. And then after that, they then go on to post-remission treatment and consolidation, followed by maintenance. Next slide. And so this is a phase 3 design. The primary endpoint here is overall survival. The expected enrollment is 330 patients, and the statistical analysis is shown here. So it's thought this will take about 5 years to accrue. So far, there are about 7 patients that are accrued, and I believe 9 patients were screened, but I believe the accrual so far is 7 patients so far. Next slide. And then another trial that Kristen and I are representatives from the adult cooperative groups This is actually a children's oncology group study, which is AALL1832. This is a phase 2 study of blinatumomab in combination with nivolumab for patients that are less than 31 years of age with first relapse. So the thought behind this trial is that increasingly the mechanism of resistance has been thought with blinatumomab has been thought to be due to endogenous T cell factors, including CD8 positive T cell exhaustion. And so there's some evidence that checkpoint inhibitors can overcome T cell mediated resistance to blinatumomab. And so Stacy Cooper is leading this trial. We can go to the next slide. And so really, this trial has 3 arms, but for the purposes of the adult group, we would really only be focused on this first arm, which is looking at either blina versus blina nivolumab. The other arms are for younger patients. And so this basically is a randomized study of blinatumomab versus blinatumomab nivolumab for patients 18 years of age and older, as mentioned, because this is a COG trial, it's going up to age 31. Next slide. So the group 1, which again is the cohort we would be accruing to, would be up to 2 cycles of immunotherapy, and patients are allowed to then go on to transplant. There are 132 evaluable patients in this arm. So far, this trial, I think, has been slow to accrue within COG, which is one of the reasons they had this endorsed by CTSU. For the adult group. So far, I think there only are about 22 patients that have been enrolled to date for all 3 arms. Next slide. And then the next trial that was opened fairly recently is S1905. This is a trial that Nick Papadakis is the co-chair with me, looking at OBI 3424 in relapse refractory T-ALL. Next slide. So basically in this trial, we are looking at, so AKR1C3 is an enzyme that is preferentially expressed in T cell ALL and expressed at low levels in other tissues. And so there has been data in ALL xenograft models, particularly T-ALL xenograft models, showing that one drug called PR104 demonstrated significantly increased activity against T-cell ALL. OBI 3424 is a newer drug that's been developed, and basically this drug is activated through this AKR1C3 pathway. So we can go to the next slide. And so OBI 3424 is an alkylating agent that is cleaved in the presence of AKR1C3. And so this cytotoxicity of the drug has been evaluated in both leukemia cell lines as well as PDX models and shown encouraging activity in the lab in both of those situations in T-ALL. And so that's kind of the background that led to this trial. Next slide. And so this is a phase 1/2 trial. The drug is administered on days 1 and 8 of a 21-day cycle. We're doing a standard 3x3 dose escalation. Patients, if they achieve a remission, are able to go on to transplant. But if they're not going to go on to transplant, they are able to receive a maximum of 17 cycles. And much of this dosing was based on the solid tumor trial. So we actually waited to open this until they were at a higher dose in the solid tumor trial, because we knew that with leukemia, we probably would need a better dose to achieve any evidence of activity. Next slide. So again, primary objectives are to look at the maximum tolerated dose. The phase 2 really is to look at response rate. And then the secondary objectives are shown here. The other thing I want to mention, Cecilia Jung, who's at Fred Hutchinson, has done just an absolutely phenomenal job of helping us with lab correlatives, looking at AKR1C3 expression by different methods. And so that part of the study has really, I think, been very critical. Next slide. And this is just kind of for interest. So we've enrolled two patients to date in this trial. This is from Cecilia, from the first patient that was enrolled, just showing AKR1C3 by immunohistochemistry. So you can see this patient had very high expression. Next slide. So now I'm going to turn it over to Kristen.
Hi, everyone. Thanks for joining. We're going to talk about the trials that we've been developing with the working group for ALL. The first trial we'll talk about is Ellie's trial with inotuzumab plus mini hyper CVD versus augmented or dose attenuated hyper CVAD. And then Angelique can present her concept with the Novartis collaboration that includes ABL001, dasatinib, and blinatumomab, and then I can finish with reporting on the 2 T-cell ALL trials that are in development. So next slide, and Ellie's going to actually present his study. Ellie, I think you're muted. There you go.
Do you hear me?
Yes.
Okay. Thank you, Kristen and Yujari, for all your hard work. And I want to thank Steve and Harry for the leadership and for allowing me to be part of this very exciting journey in ALL. And after working with our group, we finally get to the point to have a randomized trial, which is a phase 2 trial comparing mini CVD with inotuzumab to those are just hyper CVAD in patients who are 50 years and older. And the rationale is very simple. Older ALL patients have a very poor outcome. We have no standard of care. These patients, they have a high risk in CR and resistant disease, and therefore there is an unmet need. At MD Anderson, we led a phase 2 study with very promising results with the mini CVD inotuzumab in older patients with ALL. Next slide, please. Okay, well, this is the experience we published in Lancet Oncology a couple of years ago, where we treated 60 years and older patients, median age 68, with the combination of mini CVD and inotuzumab. This combination was found to be effective with a high response rate of 98% and 96% MRD negativity, with a median PFS of 35 months and a survival at 3 years of 56%. Of note, age is a risk factor for veno-occlusive disease, and the drug was found to be thrombocytopenic. Therefore, we amended the study to lower the dose of inotuzumab in order to decrease the rate of VODs. Next, please. So here's a study design that I'll co-lead with Marlies Laskin from Dana-Farber in our Alliance group, a trial. And as Anjali mentioned, we took 50 years and older, and then the Alliance trial will cover up to age of 50. These are newly diagnosed patients having up to ECOG of 2. Can you go back, please? Okay. Who don't have normal organ function will be randomized to either mini hyper CVD inotuzumab, or those are just hyper CVAD as induction consolidation followed by maintenance with pomp. And PIF will be certified by age, 70 years and older or younger, and I will show you why, as well as by expression of CD20 in order to decide on rituximab as an additional therapy. Next, please. So here the design, the schema of the trial, where we aim to, for up to 70 years and younger, to give mini CVD inotuzumab up to 8 cycles of chemotherapy followed by pomp. Of course, patients who, and then for the standard of care group, will receive hyper CVAD up to 8 courses followed by pomp. Patients who are 70 years and older can get 2 courses of chemotherapy, A1, B1, and then move to pomp maintenance therapy. For those who are younger, but then if they have a myelosuppression problem, they can move to maintenance earlier. We will get bone marrow done at the baseline, and we'll send the sample to NGS to get the barcode at the beginning. We will have a bone marrow done after cycle 1, which is between day 21 and 28, and we'll repeat the bone marrow after cycle 2, and that will serve for our primary endpoint, which I will come to mention in the next slide. And of course, we will repeat the bone marrow before starting our maintenance treatment. Next, please. So here the dose of the chemotherapy, the mini CVD. What we did, we reduced anituzumab dose from 1.8, which is the approved dose, to 0.9 cycle 1 and 0.6 cycle 2 to 4, given in a fractionated fashion. This will allow us to give up to 2.7 milligram per meter squared, and we're confident at this dose we're below what can induce veno-occlusive disease. Of course, in this group of patients, we will have less transplant because of the age, and therefore, the VOD will be less of importance. No intracyclines given, and the chemotherapy are reduced in a major way, cyclophosphamide by 50% from the standard dose, and so is the case for dexamethasone. And for methotrexate, we have even further reduction from, for example, 3 grams fosfatidine to 0.5, and methotrexate from 1 gram to 250. So as you can see, the dose are really reduced, and with the mini hyper CVD, usually we do not anticipate myelosuppression. Next, please. Here's the hypercVAD dose, and I will not go into details, but just want to highlight that the dose will be adjusted based on age of the patient and their performance status in order to avoid toxicities in this group of patients. For example, if you look at the intracyclines are heavily reduced by 50% for older patients and less for intermediate group of patients. So watch carefully for those adjustments. Next, please. So the primary endpoint is the 2-month event-free survival in a landmark fashion. So essentially, patient will get 2 cycles of treatment and will be assessed at that time. And event is defined as MRD negative remission. So essentially, we're integrating a new concept in assessing event-free survival because we know MRD positivity is a major predictive for poor outcome, and we know that the FDA approved the BLAST trial based on MRD positivity as well, and MRD can be considered surrogacy for survival. Therefore, event is defined as MRD negative remission after 2 cycles of treatment, which will be 2 months. This is why it's very crucial to assess at 2 months. And the threshold for positivity is 10 to -4. And this will be standard of care by flow. Any relapse, molecular relapse or morphologic relapse will be considered an event. Death from any cause. These are the events we'll capture to define our event-free survival in this trial. Next, please. So when do we assess for response? Of course, as I showed you after cycle 1, A1, and then B1. If patient is not responding after cycle 1, can get a cycle 2. If patient is not responding after cycle 2, will be taken, morphology will be taken off study. If MRD positive, will get to Blina. Patients who is positive at cycle 1, positive by MRD, can receive to cycle 2. The endpoint is essentially at cycle 2 to assess for MRD. If patient is MRD negative, can stay on therapy. If patient is MRD positive, can be taken off study to receive standard of care, which is blinatumomab. Of course, anytime we have a relapse, patient will be taken off study. Next, please. So for the 70 years and older, we know with the chemotherapy, even with the mini CVD, hyperdose, adjust hypercVAD, the tolerance is very poor. So we designed a trial to give 2 cycles for 70 years and older and move to pump. Of course, if you have a transplant option, can be considered. Otherwise, patient can receive up to 8 cycles and then pump maintenance. Patient can proceed earlier to pump if they have a problem with tolerance and myelosuppression. Next, please. So the statistics where we work with Vivian from the Alliance Group, and before I'd like to thank for all her work my dear friend Megan and Anna from the SWOG, where we went to a small phase 2 trial from a phase 3. We were very ambitious. We went to phase 2 study, with the primary endpoint will be 2 months EFS. We hypothesized that with the hypercVAD it's a 40%, and we can go up to 65% at least, which is 25% improvement. The study had 85% power to confirm these results. Of course, will be early stopping rules for futility or lack of efficacy observed. Next, please. So the accrual was based on the ECOG 1910, where they accrue 4 patients per month. In our study, it will be widely open across the different sites, and we will enroll 70 years and older. We think will be high interest because there's no competing trials, and therefore we think we can accrue around 6 patients per month, and that will be feasible. Therefore, with a goal of enrollment of 90 patients, we estimate this study will take 15 months to accrue the patients. So therefore, it's a short time study that hopefully will get us to the next step to establish the new standard of care where we can go for a big phase 3 randomized trial. Next, please. So the secondary objectives are in small font here, but essentially we look at the response rate, we look at the MRD negativity at different time points, we look at the event-free survival as original EFS, not the MRD. We look at the safety, mainly the veno-occlusive disease. And then something important here, we're looking at the geriatric assessment and frailty in this older group of patients and impact on therapy and outcome. Next, please. Before I let this to Dr. Anjali, I'd like to add for this trial, we're exploring 2 correlative studies. We're looking for MRD by NGS, which will be done. We will compare to flow, and we do blood versus marrow on NGS. This is really important because then we can have a new tool to assess for MRD using NGS. And the second, as I mentioned at the end, the genetic tool to assess frailty and impact on therapy and outcome. Thank you very much for this opportunity. Happy to take questions. Anjali and Kristen, back to you.
So this is Anjali. I'm going to talk about this next one briefly, very briefly, because I want Kristen to really be able to focus on her study, which is really exciting. So Michaela and I are working currently on a phase 2 study of asciminib, which is the ABLE-001. Or TKI that's being made by Novartis in combination with dasatinib, followed by blinatumomab in older patients with Ph-positive ALL. So this is going to be a trial that we're working on as part of the SWOG CTP with Novartis. So we have a concept that's drafted and are kind of in the early stages of working on this. So I'm going to let Kristen go ahead and take over to talk about her upfront T-cell trial.
Anjali, thank you for that quick announcement about this effort. As Kristen's taking over, though, I just want to make the comment. I got an email 30 minutes before we started from Kathy Albane, who is our SWOG leader of the Clinical Trials Partnership. And she received an email, I think it was today, but very recently, from Novartis stating that as the agreements stand right now, Novartis agrees to the agreement for the Clinical Trials Partnership, and they are circulating it at Novartis for signatures. So it looks like this is going to happen. So you're ahead of the game. We will shortly have the agreement, and we'll be able to formally send them this concept for their review. So thank you.
Great, it's wonderful news.
We have the next slide. You can. This is all about Anjali. Do you want to discuss this, or do you want to just get to the T-cell trial?
Let's go to the T-cell trial. I think that'll be good to talk about, Kristen.
Next slide. Next. Okay. Thanks for letting me talk about this trial. I know we've talked about it at some of our leukemia meetings before, but just as background, we haven't had a T-cell trial in quite a while. So as you all know, T-cell is curable for about 80% of children and approximately 70% of the young adult patients who mainly are afflicted by this with contemporary frontline chemotherapy regimens. But approximately 30% of our young adult patients will have subclinical or MRD following induction and consolidation therapy. And we know that this is an important risk factor for relapse, and that relapse TALL is incurable for most adult patients with an OS following relapse of less than 10%. And there's no real effective salvage chemotherapy or novel immunotherapies or targeted therapies exist at this time. There is preclinical and early phase clinical data that support the rationale for targeting survival pathways in TALL. Next slide, please. This slide serves just to remind you about the data from the CALGB 10403 trial that did include T-cell ALL patients, and that the outcome with the pediatric inspired regimen did not differ between B and T-cell phenotype. So this makes up the majority of our backbone. Next slide. And this slide just is a reminder of the COG large AALL 0434 study that incorporated nelarabine into their frontline regimen. And this data also informed the design of the standard chemotherapy backbone that we are using, which is essentially the CALGB 10403, but then adding nelarabine starting in consolidation. There are 6 5-day courses of nelarabine at a dose of 650 milligrams per meter squared per day. Next slide, please. So here's the schema for SWOG 2113. There's the screening, enrollment, and baseline MRD assessment to identify the phenotype. The MRD assessments will all be done in Brentwood's lab, and then induction will be randomized to the CALGB 10403, whatever backbone you think of it as, or 0434, plus or minus venetoclax and low dose navitoclax. There will be a day 29 MRD assessment. However, in T-cell ALL, the MRD clearance kinetics are different as compared to B-cell. And so you don't act on the day 29 MRD assessment as long as on day 78, which is the following, approximately 10 weeks of consolidation therapy that includes nelarabine. It's the day 78 MRD assessment that predicts outcomes. So at that time, the MRD will be assessed. Patients who are MRD negative will continue on with interim maintenance with Capezi-style methotrexate and venetoclax and navitoclax, and then delayed intensification with nelarabine and maintenance. And for those patients who are MRD positive or have stable disease, they'll have an opportunity to go off trial and enroll on another trial that is being developed in the intergroup that I'll talk about in a moment, that's using daratumumab as an immunotherapy to potentially clear MRD. However, if that trial is not open yet at the time, or if patients cannot go on, they can stay on trial at the physician's discretion. This trial will also include patients with lymphoblastic lymphoma, and that assessment will be done by radiographic response, not MRD. Next slide, please. So again, there will be 2 phases to this study. There will be a confirmation of tolerability given the novel regimen that includes not only nelarabine for our young adult patients, but also the ven and the nav, which could be very myelosuppressive. And then the phase 2 portion will open to assess the anti-leukemic effect, and that will be measured by MRD and rate of disease-free survival. Next slide, please. So again, the stage 0 is just a confirmation of tolerability. There will be about 10 patients. We hope that this could be completed in about 6 months. Patients will go through induction, consolidation, and the interim maintenance blocks, because those are the 3 blocks where patients are receiving the ven at 400 milligrams per day and navitoclax at 50 milligrams per day. And this is based on the phase 1 data that was for the relapse refractory patients with both B and T cell ALL. The ven and nav will be given on days 1 through 14. And so if fewer than 3 of 10 patients don't experience toxicity, then we can start with randomization. And we will be monitoring for excessive myelosuppression, perhaps using growth factor support to limit prolonged myelosuppression. Next slide, please. Again, the primary endpoint for the stage 1 will be an MRD negative rate at day 78. We expect 30 patients per arm, so a total of 60 patients. And the way Megan has designed the studies, that we can use the patients that were enrolled in the stage 0 confirmation of tolerability phase. We expect accrual to take about 2 to 2.5 years. And there's an 80% power with a one-sided alpha to detect a decrease in the MRD positive rate of approximately 40% to 50%. 15%. And our accrual is based on the accrual from C10403. Next slide, please. And our second primary endpoint will be event-free survival. We felt that this was important. And again, we will continue on with the stage 2 design if we're actually meeting accrual of greater or equal to 2 patients per month, remembering that this is a rare disease. But we hope that in this stage 2 portion, we can accrue an additional 24 patients. That's 12 patients per arm. So we'll have a total of 84 patients. And then this will be able to provide us with power to detect an improvement of 3-year EFS from approximately 65% that was identified in the 10403 trial to 80%. It's expected that will require up to 4 years of follow-up, however, because the EFS is pretty good. And so the total design of the trial will require 7 years for completion. Next slide, please. The translational medicine objectives are MRD as measured by flow and MRD as measured by next generation sequencing, similar to the pH negative trial for older adults that Ellie just presented. And this although the MRD analysis will be done in Brentwood's lab. Next slide, please. So,
Kristen,
yeah.
before you go on, Kristen, this is Harry. Pat brings up, Stiff brings up an important question in this patient population. Will patients be able to come off study for an allograft?
Yes. Yep. So in the schema, I showed that if patients are MRD positive and you are, obviously that would be the patient population, especially that you would want to get to an allograft. And if we can get those patients to an allograft in an MRD negative state, that's why this trial was designed. So this would be this is a phase 1/2 single arm study to determine the rate of complete MRD response following daratumumab for adult patients with T-cell ALL and CR1, but with detectable MRD. So we know from the 0434 study, which did include patients up to age 30, but it was only 7% of the total patient population, but the MRD rate was approximately 30%. And measurable MRD at that day 78 time point was associated with inferior EFS and OS as compared to undetectable MRD in 0434. And that even included ETP patients, so high-risk patients who normally you think of have very poor outcomes. If these high-risk patients were MRD negative, they had equivalent outcomes to those patients who, non-ETP patients. And also the NOFO 2008 study revealed about a 30 to 40% MRD positive rate as well. And in that study that was published this year in 2020, they were able to show that there was a much higher risk for relapse for patients who were MRD positive with a hazard ratio of 3.1, and that most relapses did occur while patients were on treatment, and the median time to relapse was 1.1 years. So we know that this is a risk factor for relapse, and this is what we're trying to fix. So next slide, please. So Dave Ticci's lab has shown that CD38 is expressed on T-cell ALL and that it retains its expression despite chemotherapy exposure, which is important in the design of how this study. And in the models, in the small studies that have been done, daratumumab plus some low-dose chemotherapy has cleared MRD, again in these very small studies and case reports. And CD38 expression did not correlate with response. So as long as you had some CD38, you were able to get the drug. So next slide, please. So the proposed schema for the Dara MRD trial, again, the eligibility would be patients with T-cell ALL in first or later hematologic CR, persistent or recurrent MRD after a minimum of 2 blocks of intensive chemotherapy, or developing MRD following an allo. So the Dara will be given at the myeloma dose, which is 16 milligrams per kilo. It's given IV weekly for 4 doses. We thought that the total dose is recommended. It's an 8-dose cycle, but we we felt that the group felt that it would be important to do an MRD assessment after 4 doses to make sure, especially for those patients that have persistent or recurrent disease, that they weren't taking off, so that we have an early look. But anyway, so patients can continue on if they're MRD negative to receive 4 more doses and then have another assessment and then proceed on to transplant. If at that first MRD response after 4 weekly doses, they still had significant MRD, then you can add some chemotherapy, which we think would be a Capezi methotrexate. The primary endpoint of this trial is MRD response after 1 total cycle, which is 8 doses of Dara. Next slide. In the interest of time, I don't know if Steve, if you want me to go through all this, but
no, I think I think it was just important to show that concept, and we'll hear more about it as it comes through, obviously. So thanks for that great overview of ALL. As you can see, some exciting times ahead towards a future of chemotherapy-free treatment, at least for elderly ALL, we hope. So we're likely going to run a bit over, so I hope folks can stay with us perhaps to about 1/4 the hour. I'd like to turn things over next to Jerry Radich to update us on translational medicine efforts in the committee.
I believe you have my slides. Hi, everyone. So, Steve, because of time constraints, do we have some other clinical trials to go through?
We have Kendra's just to give us a brief update on her CML trial.
Why don't we do that, and then however much remaining time you want me to do, I can modify mine for that.
Okay.
Because originally you had me down for 20. Obviously, that's not going to be appropriate. So why don't we finish off all the clinical stuff, and then people, you can tell me how much time you want, and people who want to stick around can stick around.
Okay, let's
Mine will
get
take
to...
truly 2 minutes.
Go
It'll
ahead,
be the
Kendra.
shortest of all.
We'll go through, and then we'll go to Gail on patient advocacy. So Frank, if you can skip through to the end of the deck.
That's the only good shot anyway.
Yeah, that was a beautiful picture. Okay, perfect. Kendra.
All right. Thanks. This is truly a 2-minute update. So S1712 randomized phase 2 trial of TKIs plus ruxolitinib in chronic phase CML. Currently ongoing. I won't give the background because I think at this point people are familiar with the background. It's been open for 2 years probably now. Next slide, please. So the schema for this study, this is again randomized phase 2 trial for chronic phase CML patients. As it stands right now, eligible patients are those receiving 2nd generation TKIs, nilotinib, dasatinib, or bosutinib, with no history of TKI resistance. They must have achieved at least a complete cytogenetic response, meaning PCR less than 1% on the international scale, but still have detectable BCR-ABL. And they must have been on treatment for a minimum of 12 months and their current treatment for a minimum of 6 months. They're then randomized 1-to-1 to remain on their current treatment or remain on their TKI plus ruxolitinib 15 milligrams twice daily. They then remain on study treatment for a total of 12 months, at which point they come off study. While on study, they're getting PCR checked every 3 months, as we do for standard of care. And the primary endpoint of this study is rate of MR 4.5 at 12 months. So total enrollment, the goal is 74 patients. So far, we've enrolled 52. You can see the allowable doses of all TKIs listed there at the bottom. Next slide, please. You know, we are certainly making some headway with enrollment on this study. I think when we started to allow bosutinib and when we adjusted some of the doses of TKIs, that certainly improved enrollment to some extent. But we're making a couple of other changes that I think will help as well. And I think we'll probably be able to finish enrollment much sooner once the next amendment comes out. It was recently submitted for approval. And that is that we're going to start allowing imatinib treated patients as well. So that amendment, like I said, it's been written. I believe it was submitted for approval. If not, it should be submitted any day now. So that's coming soon. And then the only other change in the amendment is wording in the eligibility to make it more clear that really the goal is that patients can't have a history of resistance. Right now, it kind of sounds like what we're looking for are patients who are intolerant to prior therapy. That's not actually the goal. The goal is just no history of resistance. And typically, if they've been switched from one treatment to the next, it's either for resistance or intolerance. So the way that it sounds now is that they have to have been intolerant. That's not really the case. It's just that they cannot have been resistant. So we're clarifying that and then allowing imatinib treated patients. And hopefully we can finish up enrollment in the next year and then get to the primary endpoint within a year after that. Next slide. I think that was actually the last slide. Yeah, so there we have it.
Great, thank you, Kendra. Nice uptick in enrollment over the past year. Frank, if you could scroll back a few slides to the beginning of Gail's presentation. Beautiful. And Gail, we'll turn it over to you and Bruce Wright. Just need to unmute yourself.
Sorry about that. I was saying such important things there. Anyway, thanks for the opportunity to speak for just a few minutes. I do also promise to be brief, but I think the first thing I really want to do is just to acknowledge the tremendous and exciting work that happens in this community, in this committee. As I always write on the triage slide, I always start it with from a patient and family perspective. Well, from a patient and family perspective, I just really want to say many thanks to all of the physicians for their dedication and commitment to patients with leukemia. It warms my heart. Anyway, just a few updates about the SWOG patient and community advocates. In 2019, the SWOG Patient Advocate Committee launched the Community Advocate Program. Since that time, has brought on 10 new advocates. We're going to hear from Bruce in just a second. And what's been really great about it is that they bring diversity and close connections to a variety of communities. It's still in the developmental stages. It hasn't been fully rolled out, but it will increase the number of advocates from 22 to 32, and that's pretty profound. So stay on this slide just for a sec. Just to point out that none of us are physicians. I think you all know that. Experts a different way. I like to think we bring a different expertise to the table, and that is the patient voice. So it's really a strong and powerful patient voice. Research advocates know trials and the questions we need to ask, especially when it relates to things like science and statistics. And we either have extensive experience in trial development or we're trained in trial science. I think most of you know, in my case, I was with the Leukemia and Lymphoma Society for 20 years. On the other hand, community advocates know their communities. They really know the concerns and the priorities of the patients that they represent. And here's a listing of the 10 communities. Recently, we have excellent representatives in each of those. The next slide, please. Just a couple of key differences. Research advocates are embedded in one community, committee, I keep calling them committee, such as myself, embedded in the Leukemia Committee, and community advocates can be called to work on a project or offer advice across any committee, and so they can really at any time work with several. So you can see that the numbers again will be different. There's 20 advocates on the SWOG committee and with an additional 10 working right now across SWOG. Next slide, please. Just a review of these are the big questions that we ask when we're looking at a trial, everything from how important is this trial all the way to are there any patient-related issues that should be considered. I've highlighted and bolded in black some of the areas that we're currently focusing on in the Patient Advocate Committee, and that would be certainly broadening inclusion criteria. Some of you have probably heard me ask that question. Heavy focus now on diversity and inclusion. A lot of focus right now on patient-friendly informed consents. We look at the issue of financial burden for the patient and his or her family, and also quality of life issues, another big area for us. So, and then my remaining minute and a half, I would like to just introduce Bruce Wright. He's the community advocate.
Gail, I have to say this. I think every investigator on this call will share my feeling about this. Good luck with that patient-friendly informed consent, a major barrier to getting patients on studies. Godspeed, make it happen.
God bless you. Yes.
There's a new criteria coming out. I would share my frustration too, because I made recommendations. I spent a lot of time making recommendations, and I was said, no, it has to stay this way. No, it has to stay this way. But you know, hopefully we're going to be putting information into what we like to call digestible chunks. So thank you for your good wishes and good luck, and I'll do my best. That's all I can say. Anyway, Bruce Wright is a community advocate for veterans issues. He's also a CLL survivor and very active with the CLL Society. And I just thought it might be interesting to have him share a little bit on his perspective. Go, Bruce.
Hi, everyone. I'm Bruce Wright, and I have CLL. And I'm the reason you all are working so that the audit of my years can be fruitful and enjoyable. Thank you. I am a participant in the CLL Society. I'm a senior support group advisor. I keep an eye on 10 support groups. We have 38 throughout the United States, Canada, and Ireland. Other parts of Europe are turning on, so we're international. I bring to you the voices of over 1,500 patients and their caregivers looking for additional information to help us live a more fruitful and enjoyable life. I am the veteran advisor for CLL Society because my CLL came from Agent Orange. Look at that. I am on the patient advisory board as a senior member, and we encourage you all to come pay us a visit. Come, I'll give you the info. Come visit our monthly meetings. I'm in South Orange County. We have 170 CLL patients signed up. What you can do to impact us. We've had Dr. Rosen from City of Hope. We've had Dr. O'Brien from UCI. We have Dr. Kipps from UCSD. And this coming month, Dr. Choi from UCSD will participate in our monthly meeting. Please help us improve the interface to help you. Thank you.
Thanks, Bruce. We'll be turning to you as we design our next upfront studies for CLL. So thanks for all your advocacy, and thank you, Gail. Well, we're saving the best for last, and for those who can stay, hopefully Frank will let us stay on for 10 more minutes to 11:10. So Jerry, take it away.
Actually, I will do this in 2 minutes without slides, and we can do data and stuff at another time.
Okay.
So the issue with the leukemia, with MyeloMatch, I can just kind of walk you through where we are in that, because that's the most pressing concern right now. So when a patient is diagnosed, they will write or they will be put on the master screening and reassessment protocol, which is basically allows us to get samples and rapidly turn them over to get their genotyping and the like done. So that's essentially the portal to any trial you're on. So that has been drafted, that's been written. The concept has been approved by SWOG and CTEP. So we're pretty much on the road there to having that done soon. The second aspect is, once that's done, samples go out into the world and get cytogenetics and genetic testing and flow and the like. As Harry alluded to, there's so much money involved in this that the NCI had to go through a different mechanism and ask their independent contractor, Leidos, to basically select places to have this done. You couldn't petition to have it done like there was no application process. They just were going to go out and find the place and see if they fit the criteria. Since I'm on kind of chairing the lab committee, I offered to recluse myself from this. That didn't work. So it looks like the lab stuff will be doing, Minfang will be doing the rapid cytogenetics. Brentwood, which was here, but now went to Children's LA, will do the flow. And the rapid genetics testing will be done Monday through Wednesday at MOCA at Fredericks, and Thursday through Sunday at our place, because we stay open on the weekends and they don't. So that's how that's going to be divided up. The machine that we're using is a so-called ion torrent machine. It's now called Genexus because it was bought by Thermo Fisher. It's a very rapid sequencer. Basically what it does is when you synthesize DNA and put on a base, you lose a hydrogen. And so it's a chemistry unit hooked up to a semiconductor. And so when you synthesize each base pair, it quickly squirts A, C, G, and T across, and whatever base emits a hydrogen ion, it's digitalized as yes or a no, 1 or 0. And that's how the whole stuff is processed. So it's really the world's most expensive pH meter is what it is, really. And so it will go from, start sample to finish in less than 72 hours. And Cecilia has been really helping us work on this, because what we have to do now, it's a new platform. And so we meet with MOCA weekly and with Thermo Fisher every 2 weeks, because we're running controls and known mutations and the like in there. And, we're trying to find out where the errors are, because sometimes calls will be made that aren't there, and vice versa. And that's either a chemistry issue or a bioinformatics issue. So we're kind of grinding through that. And so that should be done pretty soon. And then we have a phase where we found, I think, about 50 samples where we have the Illumina sequencing done on an Illumina platform. And that will be sent to both of the labs blinded to see, how we stack up. So that all is ongoing. And I think we'll have it in time ready to go for when these trials hit. That's
all right. Well, thanks, Jerry. That was the tour de force. So thanks everyone for staying with us. Just a few last words. I want to acknowledge Harry's continued leadership of the committee. We're really moving forward. And I would also be remiss if I didn't mention Rich Little. Rich has been absolutely critical, our advocate, our advisor. It's been a real privilege to work with Rich as we bring these trials forward across our entire portfolio. We couldn't do it without him at CTOP.
That's absolutely true.
Hopefully, I think we should be able to, these slides will be posted on the website and maybe even the presentation, but obviously if anyone has questions, reach out to the various folks you heard present. If anyone wants to get involved in trials, please let us know. So, Harry, I'll leave you the last word.
Thank you, Steve. That's it. Those are the last words. Steve, thanks for putting this all together. Thanks for the hard work of the entire committee. Things are really coming together. Milo Match is taking on a life. It's a real thing. Really appreciate everyone's efforts here. And, it really signifies what we are doing across the diseases in this committee is really functioning as a truly cooperative group. We've always cooperated with each other. Now we cooperate fully with all of the other NCTN groups, and we're cooperating at a much earlier stage with the NCI so that we don't waste time in developing concepts just to find out that it's not what they're looking for. They don't have the budget for it. It's not their priority. We still answer to the Leukemia Steering Committee, but by the time concepts get there, we are quite hopeful that our colleagues there will see the work that we've put in, the thought that we put into each of these concepts, and have hopefully constructive ideas in their review, but will agree with us that the basic concept is worthy of going forward. So exciting times, as Steve has said. A lot is taking shape, and appreciate everybody's time and efforts into this, into all of the activities. Thank you all.
Thanks, and thanks to Frank for the technical support. Everyone take care.
Bye.
Hi, everyone. Thank you.
Bye.
Are you still there?